Bismillahirrahmanirrahim. In this lesson, I will describe about mitochondria, plastids, endoplasmic reticulum, and Golgi apparatus from textbook pages 62 to 64. So, mitochondria, singular mitochondrion, are double membrane bounded structures found only in eukaryotes. These are the sites of aerobic respiration and are the major energy production centers. So it is also called powerhouse of the cell. The outer membrane of a mitochondrion is smooth, but the inner membrane forms the infoldings. So here you can watch the infoldings, which are known as cristae. In the inner mitochondrial matrix. So this fluid is mitochondrial matrix. This serves to increase the surface area of inner membrane on which the membrane bounded reactions can take place. The next one, plastids. Plastids are also membrane bound organelles that only occur in the cell of plants of and photosynthetic protist algae. They are of three types. Chloroplast, chromoplast, and leucoplast. Like mitochondria, chloroplast is also bound by a membrane. So here is chloroplast, then outer membrane is smooth, while this inner membrane gives rise to the sacs called thylakoids. The stacks of thylakoids is granum. Plural is grana. Grana floats in the inner fluid of this chloroplast, which is known as stroma. Chloroplasts are the sites of photosynthesis in eukaryotes. They contain chlorophyll, the green pigment necessary for photosynthesis, and associated pigments. These pigments are present in the thylakoids of grana. So pigments are present here. The next one, chromoplast. The second type of plastids in plant cells are chromoplasts. They contain pigments associated with bright colors and are present in the cells of flowers, petals, and fruits. Their function is give rise to colors to these parts and thus help in the pollination and dispersal of fruit. Last one is leucoplast are the type of plastids. They are colorless and store starch, proteins, and lipids. They are present in the cells of those parts where food is stored. The next one is endoplasmic reticulum. Endoplasmic reticulum is a network of interconnected channels. These are the channels. Here are also the channels that extend from cell membrane to the nuclear envelope. The network of these channels exist in two forms: smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum lacks ribosomes and is involved in lipid metabolism and in the transport of the materials from one part of the cell to other. It also detoxifies the harmful chemicals that have entered the cell. Rough endoplasmic reticulum is so named because of its rough appearance due to numerous ribosomes. Here you can watch different kind of ribosomes that are attached to it. Due to the presence of ribosomes, rough endoplasmic reticulum serves a function in protein synthesis. An Italian physician Camillo Golgi discovered a set of flattened sacs. So here are set of flattened sacs which are known as cisternae in the cell. In this set, many cisternae are stacked over each other, and the complete set of cisternae is known as Golgi apparatus or Golgi complex. It is found in both plant and animal cell. It modifies molecules coming from rough endoplasmic reticulum and packs them into small membrane bounded sacs called Golgi vesicles. These sacs can be transported to various locations in cell or to its exterior in the form of secretions. The next one is lysosomes. A Belgian scientist, Christian René Didieu, discovered lysosomes. These are the single membrane bounded organelles. Lysosomes contain strong digestive enzymes and work for the breakdown or digestion of food and waste materials within cell. During its function, a lysosome fuses with a vacuole that contains the targeted material, and its enzymes break down the material. Here is Golgi apparatus, pieces of targeted materials. Primary lysosomes having digestive enzymes, then the vacuole and targeted material, then there would be fusion, then there would be pieces of targeted material. I have discussed about lysosomes. The next one is centrioles. Animals and many unicellular organisms have hollow and cylindrical organelles which are known as centrioles. Each centriole is made up of 9 triplets of microtubules made up of which are made up of tubulin protein. Animal cells have 2 centrioles which are known as centrosomes and which are located near to the exterior surface of nucleus. The 2 centrioles are collectively called a centrosome. Their function is to help in the formation of spindle fibers during cell division, and in some cells, centrioles are involved in the formation of cilia and flagella. Next one is vacuoles. These vacuoles are fluid-filled, membrane-bounded, single membrane-bounded organelles. Cells have many small vacuoles in their cytoplasm. However, when a plant cells matures, its small molecules absorb water and fuse to form a single large vacuole in the center. Cell in this state becomes turgid. Many cells take in materials from the outside in the form of food vacuole and then digest the material with the help of lysosomes. Some unicellular organisms use contractile vacuole for the elimination of wastes from their bodies. Now it's time to discuss the home tasks for the day 2. You have to read and memorize the concepts about mitochondria, chloroplast, smooth and rough endoplasmic reticulum, Golgi apparatus, lysosomes, centrioles, and vacuole from textbook pages 62 to 65. You have to draw mitochondria, chloroplast, smooth and endoplasmic and rough endoplasmic reticulum, Golgi apparatus, lysosomes, centrioles, and vacuole. Thank you and have a nice day.